Yeah, I think we're live. Okay, I think we're live. Good. So maybe we can get started. So welcome everyone. It's my pleasure to introduce Dr. Hari Sadasivan today. Hari is a member of technical staff in the AMD AI group, a faculty member part-time at the University of Washington, Seattle, and a senior member of IEEE. Hari's research focuses on three critical and emergent technologies that are identified as significant for the US national security, advanced computing for artificial intelligence, and biotechnologies. At AMD, Hari leads the AMD Center of Excellence in AI at University of Washington, Seattle, and co-founded the AMD Life Sciences Group. Hari received his PhD and master's in CSE from the University of Michigan and Ann Arbor. Hari is a technical reviewer with several IEEE ACM venues, including the longest, oldest, and the most prestigious formats. Hari has previously worked with NVIDIA and Samsung R&D. And I guess most of us know several of his works because we're also very interested in particular directions that he's working on, such as long read alignment in general genome analysis. So with that, please take it away, Hari.
Thanks, Jan, for the introduction. It's my honor to present at the Safari Group. I have followed some of the seminars here and also been a big fan of Honor's public lectures. I think it was really useful for me towards the beginning of my PhD. So welcome all. Today I'm going to talk about accelerated systems for long read alignment, and my name is Hari. And if you have any questions throughout the seminar, please feel free to stop me and ask them right away. Or if it's something that needs a detailed discussion, please feel free to hold on to it until the end of the seminar. So a little bit about me. I think Jan covered most of it. So I work at AMD and also at the University of Washington. So my work at AMD is mostly in the AI space and also in the life sciences space. And at the University of Washington, I design and teach courses on GPU programming, particularly in the hardware and also more recently in the software space. And I do also contribute to the MLSys courses there. Other than that, I'm an active member in the IEEE and I'm a motivational speaker for IEEE HKN. And I did my PhD and master's, as Jan said, from the University of Michigan, Ann Arbor, and I've worked in a few other companies as well. So if anybody is interested in any of these topics that I talk about, please feel free to reach out to me. I have my email and home website information at the bottom of the slide. So I'd like to start today's talk. I know some of this content might be familiar to you. I've seen some of this contents discussed in some of the seminars, but I'd like to put all of those together in line with how I'm thinking about this or how some of us at AMT are thinking about it. So genomics is seeing a rising importance, as you probably know. The cost of DNA sequencing is falling much faster than what Moore's law is falling. I mean, if you're comparing to the rate of fall of rate of Moore's law, the cost of DNA sequencing is falling too. So to put this in context, if you see the cost of DNA sequencing at the beginning of 2000s, it would typically cost you like more than 10 million to sequence, or closer to a 10 million. I mean, in terms of order of magnitude, to sequence a complete human DNA. But currently, you can sequence it. I think it's very recently gotten to the point where you can sequence it within a few $100 or $200. And why is this important? This is important because of multiple reasons, but the three main reasons that I like are cancer, metagenomics, and rare genetic diseases. So what this means is that we can use DNA sequencing to study and understand if you have cancer, what type of cancer, and if you can understand the diversity of the microbiome in a sample, like let's say human saliva or gut, and or maybe understand genetic conditions that might be rare to detect and in unborn babies or newborn babies. And these are all, I feel, very revolutionary applications that we gain out of genomics and DNA sequencing. And the importance of DNA sequencing is identified by the US government as pertaining to the national security. It definitely comes under the category of biotech, along with a lot of other stuff. And DNA sequencing also contributes to the US GDP. Just from to put some number into the perspective, I mean, if you were looking at the US blue economy, it's again really important to understand the ecosystem. So you need to understand the microbiome, or the diversity of the microbes in the marine ecosystem. And this is important for several reasons. Feel free to look that up. But this alone contributes approximately $300 billion to the US GDP. So genomics is not just limited to healthcare, it also has applications in very other fields. And why is this important to us as computational engineers? And the reason is genomics is proposed or soon to beat astronomy and YouTube in terms of the amount of data produced per year. And the data is expected to be around 40 exabytes per year. That's supposed to be the data generation towards the later part of this decade. And we have already seen this explosion right from when COVID hit us. So right from then, people have been sequencing different strains of the virus itself from different parts of the world, and we've been seeing this kind of explosion. And this is interesting to us as computational engineers because this poses us a storage and computational bottleneck, which is a problem for us to work on. And it's a very, very important problem for us to work on. Please feel free to read our paper, very recent paper in collaboration with Professor Onur Mutlu. So this is, we have a lot of people working on I mean, I'm thankful to a lot of people from AMD and also some of our external collaborators who worked on this paper. We recently presented this at the IEEE. If you feel free to read about the rising importance of genomics and how we propose to encounter this problem on this paper. And next, I would like to introduce you to the revolutionary handheld sequencer MinION. Some of you might be already aware of what the MinION does, but I'd like to summarize what it does. So MinION is again a portable DNA sequencer. It's handheld. It's very inexpensive. It costs like approximately $1,000 for you to get a MinION flow cell. And it's the attractive feature of the MinION that makes it stand out from the other sequencers is it can do real-time sequencing. So as the DNA passes through the pores or the nanopores, which are like really small pores on a membrane on the sequencer, you can see or you can observe the characteristic current fluctuations that the nanopore detects from the DNA passing through it. And this signal can then be processed by a pipeline of softwares for us to understand what DNA we are sequencing, and then try and map this DNA to some piece of golden reference that we have and try to figure out where this DNA came from. I mean, the software pipeline could look very different depending on the application, but the bad news is that the MinION lacks edge compute power. And this really is not good for the MinION, especially given how portable the sequencer is. And imagine having to connect it to a desktop or a cloud, or even send the data to a cloud server just to process the high throughput output from the sequencer. That doesn't look good. And if you look at the applications of the MinION, there are, I mean, apparently 1,000 different applications that are understood as by the manufacturer Oxford Nanopore. But I'd like to start the discussion by starting with pathogen detection, because I feel all of us were affected by SARS-CoV-2, and it is really important for us to be able to understand and detect a pathogen and test for it. So if you look at pathogen detection, the problem is very similar to searching for a needle in a haystack. The reason being the presence of a pathogen or a microbe in a human sample is going to be very, very small. It can vary from 1% to 0.1% in out of 100%. So we are really searching for a needle in a haystack. And this takes often, as the name sounds, it takes a lot of time. And we have identified a few bottlenecks in some of these pipelines and come up with solutions. So for the pathogen detection problem, to effectively solve it, first we came up with the hardware software co-design. We called it SquiggleFilter. SquiggleFilter is a reprogrammable ASIC that runs custom software. I'm sorry. And we won the IEEE micro topics with honorable mention in 2022 for SquiggleFilter. And then we realized that an ASIC is not really democratized. It's not really accessible to everyone in the world. And that's when we decided to move the SquiggleFilter implementation to a GPU. So DTWax is our SquiggleFilter software optimized for GPUs. This was done in collaboration with NVIDIA. And finally, I'd like to highlight one work that I did at NVIDIA, and also like was able to open source it and improve it from AMD. So the idea is, beyond pathogen detection, you have like 1,000 different pipelines that can go with MinION, right? So and there's a software that helps you map the DNA to a reference, and this software is used in all of these pipelines. And we call this software Minimap 2, and Minimap 2 has a performance bottleneck that we identified, and that we solved on GPUs. And from NVIDIA, the software was closed sourced, and we call it MM2AX. That is now sold as part of NVIDIA Parabricks in their Minimap 2 beta version. And unfortunately, because it was not open sourced, it wasn't able to benefit the community. And that's why after I joined AMD, we decided to, work on accelerating and open sourcing Minimap 2 on GPUs. And that is what we call MM2GB. And the good news is we have MM2GB being used in a clinic for cancer detection right now. We'll talk about it briefly at the towards the end of the presentation. So these are the three main solutions that I'd like to discuss. Some of these solutions are still being repurposed for different applications. I'll talk about them as we go about it. Again, these are some of the performance numbers that we measured for SquiggleFilter, DTWax, and MM2X and MM2GB. And all of these works are published except for MM2GB, which is accepted at the ACM BCB conference. So a quick recap to our MinION. Although it is very convenient and inexpensive, it lacks compute power. And if someone is unfamiliar with the long-read sequencing technology, here is a brief summary of what it does so that we can put MinION in a better context. So DNA has many repeating regions. Imagine the longer, the more complex the organism, the more repeats it would have. That's how I approximate the concept in my mind. What that means is, imagine long homopolymer strings of A's, G's, C's, or maybe repeating pairs of bases like AG, 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 etc., or something like that. And DNA, when you do a wet lab preparation, just to prepare the DNA using different chemicals and stuff like that, before you load the DNA onto the MinION, this wet lab preparation is a requirement. And while doing this, the DNA gets fragmented into tiny pieces. So this is not something that we desire, but we really have to go through it as an artifact of the wet lab preparation process. And this is bad. And prior technologies could only sequence short, accurate reads. So the prior technologies were really accurate in sequencing these short reads, but it was limited to just sequencing short reads. But what we desire is in fact long reads, and so that we can solve the puzzle of assembling, imagine a very long DNA, like human genome, very accurately. So I'd like to draw an analogy to a jigsaw puzzle solving. So if you have larger pieces on a jigsaw, it's easy to put them together and solve the puzzle. But if you have tiny pieces and all of them coming from the sky, it's really difficult to understand to what part of the sky these tiny pieces in a jigsaw puzzle fits in. That's the analogy I have in my mind. And long reads, the other problem is long reads are really noisy. So although we like long reads to begin with, long reads tend to be noisy. And this noise in the long reads affects our software pipelines in terms of, let's say, if we have to detect if we have cancer, if somebody has cancer or not. We need to denoise this signal to some extent before we can answer the question. And so MinION is this long read sequencer. So long reads are good to solve the puzzle of the jigsaw puzzle of putting together a DNA. But we have to go through this whole denoising process through the softwares. But the good news is, over the years, just like how Moore's law gets us more transistors on a chip, the accuracy of the software that does this denoising has also been improving a lot. So I would say we are currently very close to or almost on par with short read technology in terms of the noise level in the reads, which is very good. And imagine it'll just keep improving, or maybe if there's any margin, it will just keep improving over the next few years. And let's add some more background to the MinION sequencing pipeline if someone is not familiar with it. So you have the DNA reads that come from a wet lab preparation. So imagine you take a biopsy or a sample from a living organism and then make it go through some processes so that you prepare the DNA after extracting it from the cells. And then you prepare this DNA in a way that they can go through these sequencers effectively, so that you are able to guide the DNA to pass through the sequencer. And once the DNA passes through the sequencer, or the sequencer has begun sequencing, you observe characteristic distortions in the flow cell of the sequencer. And these characteristic current distortions are what we call squiggles, and they are typically very noisy. And therefore we use a deep neural network. We call it a base caller. Over the years, the base caller has evolved. Oxford Nanopore used to, I think now they are on the fourth fourth or fifth different type of base caller over the few years. And previously it used to be called Guppy. Now they have moved on to something called Dorado. And Guppy or Dorado, what it does is like it denoises the squiggles into AGTC bases that we can understand. So this is the output of the base caller is human readable and something that we can understand. But the problem is the output of the base caller is still fragmented. I mean, I told you how the DNA gets fragmented in the wet lab. So how do we compare the small fragment of a DNA? Imagine like 1,000 to a 10,000 base, and how do we figure out where this maps to in a 3 billion long reference, which is your typical human reference? That's where we use a software called Minimap2. In simple terms, Minimap2 is an approximate string matching software. So what it does is it tries and maps this a long noisy string. So I say long because the length of the sequence output can vary from a few hundreds to maybe even a million bases in some very particular scenarios. And Minimap2 tries and do an approximate match of this read or query to a reference DNA. So we can program Minimap2 with human reference I mean, there are different sets of human references that researchers all over the world have tried to come up with, although they do have systemic biases in them. It's something that we can do, like program Minimap2 with some reference DNA. And then we can, maybe it could be the individual's reference DNA, it could be a global reference DNA. It all depends on how you want to set up the pipeline. But the outcome is you try and map the input query or the reads to the reference DNA and then figure out where these reads are mapping to in the reference. That's why Minimap2 is used. And once you map the reads to the reference, you could typically pass it through a variant calling pipeline. So maybe you need to correct for some errors, or it could be as simple as looking at a consensus of a position and then looking at if you have a mutation or not based on a vote. So at this stage, the outcome that we expect to see is maybe you need to look at all over your human DNA, and I'm just trying to abstract it at this level, and then come up with all the set of mutations that you have across the whole human DNA, and that would be your set of mutations in a variant calling pipeline. And in different pipelines, maybe sometimes you don't have a reference DNA to work with, and then this pipeline will look slightly different. And coming back to a problem of pathogen detection, microbes are only less than 1% in a human sample. And out of this, you could say only 0.1% is typically SARS-CoV-2 if you ever have SARS-CoV-2 infection. Or maybe if you're looking for one species, one particular species of a microbe, it would be around like much lesser than 1%. And that's why we call this a needle in a haystack problem. And wet lab microbial enrichment is what traditionally researchers used to do. And the process here is that we have an a priori knowledge of the microbes, and we know how to sequence them or by enriching them. So I think this wet lab enrichment depends on a time-consuming and error-prone process of enriching the presence of microbes in a sample. So that it's no longer a 0.1 or a 1% that we are sequencing. It's mostly 100% microbes that we are sequencing. So this definitely takes time and could have errors in them. And the MinION is attractive to this as an alternative because it can do real-time sequencing. And what I'd like to call as digital enrichment. You don't need to spend time enriching these microbes in the wet lab, and that is doing it in the wet lab. I think we just discussed the cons of that. So if we can get around it by using the MinION and still do real-time sequencing, that's a good feature. So if we look into it, we'll see that read until or selective sequencing is what Oxford Nanopore calls this feature as. So this is essentially a digital enrichment feature with MinION. Let's see how it happens. So as the DNA passes through the pore and the sequencing starts, you can have a software pipeline, look at this DNA real time, and then make a decision on whether you should sequence it or not. And you plan to sequence it if the DNA is something that you're looking for. So imagine a SARS-CoV-2 detection software pipeline. So if the DNA that's being sequenced is belonging to a SARS-CoV-2, you'd be more than happy to sequence this because that's the needle that you're looking for in the haystack, right? But if the DNA that you're sequencing is not the target that you want, so imagine a human DNA when you're checking for SARS-CoV-2. Typically, you don't want to spend time and money sequencing this very long DNA. So imagine the DNA was 1 million base pairs. That takes a lot of time to just go through the flow cell, and that consumes our chemical reagents as well. So that's something that you don't want to do. And so the software can convey a feedback back to the MinION, saying, hey, I don't want to sequence this DNA because I don't care about it, just eject it. So the MinION can reverse the voltage potential on the flow cell and then just eject the DNA out of the pore and make way for the new DNA to come to the pore. So this is the whole idea behind selective sequencing, and this is what I refer to as digital enrichment on the MinION. So how do we modify the the conventional sequencing pipeline for variant calling with read until? The answer is quite simple. So as I discussed, we need some kind of a feedback mechanism to the MinION to convey when we need to eject a DNA. And this piece of information can directly come from Minimap2. So remember, Minimap2 again is our approximate string matching software that tries and maps these bases or reads coming out of copy into versus the golden reference and then figures out where it goes to, right? And if it is a map to a viral reference, Minimap2 could just tell the MinION, hey, this is a map, I need the DNA. So typically if it's a map, we don't send that information because by default MinION would sequence it. But if it is not a map, that's when we need to convey the decision to the MinION so that it works against its default configuration. That is, it reverses the potential and then ejects the DNA out of the flow cell. And in only these instances, Minimap2 conveys that piece of information to the MinION. And luckily for us, in case of pathogen detection, this happens, I would say 99% or more of the time. So whenever you encounter a human DNA or something that is non-SARS-CoV-2, you are instructing the MinION to just eject it. So this saves a lot of time and cost of sequencing. Another problem with base calling or the AI, the deep neural network that converts.
Harry, maybe can I ask a question for your previous slide? Is there any delay between the time that you make the decision and send that decision to the device? So I guess there is some API over there. What do you think about basically the use of that API and the delays, potential delays using it?
Yeah, so there are I think two potential ways to get solve it, but the highlight is there is a delay, and if you use the API, the read until v2 API, and it uses Python-based handler over the base caller to get the base calling done, and then it runs mappy and to get the Minimap2 output. But this is slow, which is in fact when we constructed the baseline pipeline for this, we went one step ahead and we took the C++ based some of the files in Guppy and then like build a consumer producer system with Guppy and Minimap2 to get a more optimized version of the API. And we then try to make this decision. Even then, you were having a lot of delay, which comes from the performance of the base caller. And Minimap 2 is extremely fast. So Guppy is what typically affects the delay because it is not built to do read until, it's actually built to do regular base calling. It's not optimized to give a real-time feedback. That's one of the reasons why you observe this delay. And I'm going to talk about that in the next slide as to how Guppy or base caller is the bottleneck. And that's something that we overcome through a strategy that we discuss in the next few slides. Did I answer your question?
Yes, and so about this, maybe the particular delay about using this MinION API, right? Do you think like there is anything we can do about it as a community, basically? Or is nanopore really, or Oxford nanopore technology is really close or not really basically open to invite basically these communities to improve that API?
Okay. So you're talking about the side of the problem. So I think
Yes.
the way the data is collected from the pores and then how it's batched together so that we can feed it into the software pipeline is mostly under the control of, or was under the control of nanopore when I took a look at them. And definitely there is some amount of delay involved in how this data is buffered and batched, and I would say maybe this might be something that they're willing to look at because I know they're working on base calling on the edge on Mac laptops. So they really probably care about high throughput at that point. And maybe if you're looking on running a base caller on the edge on a laptop as it is, read selective sequence is something that can drastically affect the throughput. So I was just reading base calling on M4s is approximately 24 times faster than a x86. So to improve that number, I would imagine they would need to probably enable selective sequencing. And if that happens, I know they have selective sequencing support on the MinION, but I haven't really benchmarked it because it was after the time of SquiggleFilter. So maybe that it's good to benchmark how it performs on an M4 and then figure out the bottlenecks there, and maybe they would be open to work with the community at this point is what I feel.
Yeah, I hope so as well. Thanks.
Okay, that's great. Thanks for the question, Jan. And following on Jan's point, the problem with this feedback, the read until pipeline is that the base caller or the AI deep neural network that converts the squiggles to AGTC bases takes up a lot of time. So if you just profile the whole pipeline for variant calling, you typically see that base calling takes up 87 to 95% of the time, depending on the fraction of the virus in the sample. So this is not good because as Jan said, it's one of the contributing factors to the latency or in making decision. And the more you wait in making the decision, the more you lose out on saving the cost and time of sequencing because the DNA is not stopping being sequenced, right? It's keeping on going through the pores. So we want to make a very low latency decision so that we can eject the DNA before it gets completely sequenced. And I think again this was it's an old slide. Oxford Nanopore was planning a very high throughput in terms of the number of channels that they were planning to pack in a given area on their sequencer. This was in 2019, but often ONT puts something out and then they put it into their product much later on. And more recently, I was reading they're transitioning from a current sensing to voltage sensing. So I think they're definitely making more changes to their hardware, and by doing this, I think they were also talking about how they're planning to pack more into the same area on the flow cell. So I'd imagine something of this effect will be coming out soon. So this means that the sequencing throughput is going to go up as well. So we need a powerful hardware or a software solution to keep up with the sequencing throughput. And another problem that we discussed with the base caller, so we had a wet lab in our University of Michigan. So there we actually did some sequencing with microbial communities and human communities, human DNA. So we mixed them together, created different proportions. And then we sequenced the DNA, and then we figured out how much of the output of the sequencer is classifiable by the sequencer, classifiable by the base caller. So remember, the base caller is not really built for base calling short prefixes in the DNA. So when you're looking at selective sequencing, you're actually looking at a prefix of a whole read to figure out if the prefix belongs to a target or not. And base caller is in fact built to look at much longer reads, not necessarily the prefix. And it's one of the primary reasons why we saw that there was a lot of unclassified bases. If you look at, let's say this was the average read length that we observed in this particular wet lab experiment, based on the fraction of human to microbe in the sample, this is the characteristic plot of the unclassified bases, and we see approximately 60% is unclassified. This is because the prefix is really small, and the base caller is not effective at correctly base calling it. Therefore, Minimap2 cannot map these small prefixes to the reference. It's not the Minimap2's fault; it's the base caller's fault. So we do have a performance and accuracy problem, and performance is the primary problem that I'd like to highlight. And at that point in time, most of the accelerators were focusing on short reads, but we do have a lot of accelerated solutions for long reads at this point, especially from Honor's lab. And SquiggleFilter was the work that we came up with. This was at the very beginning of the pandemic. And we published this at the micro conference, and we got the topics with honorable mention. So the problem that we observed in 2020 was that when SARS-CoV-2 hit us, we had to spend some time designing, verifying, and manufacturing RT-PCR primers. So remember, RT-PCR was the golden standard test for SARS-CoV-2. It still is. So when you see a virus for the first time, you need to design the primers to test for that virus in this test. And these primers usually don't work in the first attempt. It's just like a clinical trial. Like, you don't get that lucky. And once you fix that, you need to fix that big supply chain that broke, because now you have a surge in demand for SARS-CoV-2 primers. Not any primers, SARS-CoV-2 primers. And once you fix the supply chain, by the time the pandemic is out of control. This is what we observed. So if we could fix that, if we could monitor the spread from the right from the beginning, we could better control the spread of the pandemic. So maybe it wouldn't have become a pandemic, it would have just been an epidemic. And our idea was distribute a SquiggleFilter hardware all around the globe, and then once you have the knowledge of a new virus, just reprogram this piece of hardware with this new virus's genome, and then SquiggleFilter hardware could potentially detect any kind of pathogen as soon as you reprogram it with the pathogen that you want it to look it for. And to recap, base caller is the bottleneck in our read until pipeline. So the idea behind SquiggleFilter is we'd like to skip base calling because it adds to the latency of the decision making. And of course, because we are not base calling, we also skip the aligner. And then we replace it with SquiggleFilter. So SquiggleFilter is a custom piece of software running on a custom hardware or an ASIC. And as it takes a look at the squiggles that come as output from the sequencer and then compares it to a target virus genome. And it performs accelerated dynamic time warping. That's the algorithm that we'll talk about that runs on SquiggleFilter to figure out if the squiggle that's input to the SquiggleFilter maps to the target virus genome. So if the squiggle maps to the genome, we'd like to sequence it because it's SARS-CoV-2, right? So that's the 1% or less virus that we are sequencing based on how infectious the sample is. And then if it doesn't map, so it's gonna be your human read or maybe a bacterial read, you don't want to sequence it, so you're instructing the sequencer to eject it. So this was the idea behind the SquiggleFilter. And how does SquiggleFilter does this kind of read to reference mapping? It relies on, so remember we're skipping base calling, right? So we need to map squiggles or electric current signals. So what we do know is that once you're seeing SARS-CoV-2 and you sequence it completely, we know the reference string. So between SARS-CoV-1 and SARS-CoV-2, there's not much difference. But as soon as we see SARS-CoV-2, sequence a reference string and then convert it into current signals. And this current signals, when we do the conversion, we just do a one-to-one map based on a lookup table. And the assumption is there's no noise in this reference string. And even if there is, the noise is very minimal. We have done a robustness study that shows that this noise is to a low margin, so that the results aren't affected. So this reference signal is then compared with respect to the query that comes out of the sequencer. So the output from the sequencer is then compared to this reference target, which is the viral target. And we do a two-dimensional dynamic time warping, which is essentially a 2D dynamic programming algorithm. Again, it's an approximate string matching, but in the current space. So instead of doing it in the AGTC space like Minimap2 was doing, we are doing it in the current space. That's where dynamic time warping comes into our help. And once you do that, so the idea is, I'll talk about this briefly in the next few slides, but we try to fill up this matrix, and then in the final iteration of the last column, we try to find the minimum value. And this minimum value will tell us the path of the map of the query to the reference. So from the minimum value, you trace a path along the minimum of all three neighbors. So you can look at all the three neighbors of a cell. I'll talk about this in the next slide, and then draw a path that tells us how this query is mapped to this part of the reference. Essentially, the idea is reference is super long and query is so small, and you need to figure out where this small query maps to in the reference. And that's what DTW helps us get done. And if you look at the architecture of SquiggleFilter, so we talked a bit about the software. We'll talk about a bit more later, but then if you look at the architecture, we'll see that it's a systolic array. So these are simple processing elements connected together with wires. And you have the query and the reference again. So how it operates is along each of the reference cell. So imagine all of the reference values are stored in one of the processing elements, right? So you have the first value stored in the P1, second value in P2, third value in P3. That's how it goes. And then we limit the systolic array for area purposes to 2000. And then if the reference is longer than that, we just iterate, do multiple iterations over the systolic array. So we have the on-chip buffers for query and reference. And then the first iteration, we try to compute the cell in the matrix. And the second iteration, both of the P, so P1 and P2 are active now. So that's what doing the computation. And in the next iteration, all three P's are active. And in the next iteration, only P2, P3, and P4 are active. And finally, P3 and P4, and the last iteration P4. So as you probably noticed, the parallelism here exists along the wavefront the parallelism exists along the diagonal of the matrix, and that is what we exploit using the systolic array. So instead of having a 2D systolic array just to waste area, what we do is implement a 1D systolic array and then have it share or stream the values between 1 through 4. That's the idea behind this implementation. And then finally, if your reference is small and you need to do multiple iterations, sorry, if your systolic array is small and then you need to do multiple iterations of the reference, and vice versa, right? If your query is large, you need to do multiple iterations. So we provision a mechanism to write values to the DRAM and read it back in certain places. And finally, let's come back a bit to the software. So with software, we did some optimizations to for performance. So we started off with the blue line, and we added some optimizations like we avoided multiplication, reduced the precision, removed the dependency. So this was a strategic decision based on the fact that the reference signal doesn't exhibit deletions to some extent. So to some extent, we know that this is confidently a noise-free signal, and this is we made this assumption for performance improvement, and combined all of these contributed to a slight drop in accuracy. So we went from the blue to the orange line, and we wanted to fix that. So to fix that, we added a scheme called match bonus. So the idea is that we approximately know the idea that OINT samples 10 times per base. So if the pore is seeing an A, the sensor would sample it 10 times based on the sampling frequency and the rate of translocation of the DNA through the pore. So we wanted to encourage the behavior of trying to map approximately 10 samples to a base, and discourage that behavior as you moved away from 10, and as you were as you are moving to away from 10. So this is the linear batch bonus. So it's like a increment to go towards 10, and and a penalty when you go away from 10. And once we put that in, we moved from the orange line to the green line. So the accuracy is now way better than what it was for the baseline.
Can I ask a quick question here as well?
Sure.
Maybe just to clarify, are you applying any segmentation technique here, or is this direct alignment of actual raw signals? But I assume since you're converting the reference genome to signals, there should be some segmentation for the actual raw signals as well. Is this correct?
So we're probably talking about event segmentation, right?
Yes. Yeah.
So we don't do event segmentation on the query. All we do is the mean and standard deviation based normalization. And we assume there are indels, insertions and deletions, and the signal and the query is non-stationary. So all these hold true. Versus if you're segmenting, your assumption is more or less the signal is not non-stationary at that point, right? We kind of approximately mapped all the 10 samples to a single event. But all these problems still exist if we don't segment. So we don't do any segmentation with SquiggleFilter.
I see. Okay.
That's where the match bonus really helped us. Yeah,
Actually, yeah, that was my follow-up question. Yeah. Because like when you said match bonus, I was really thinking about you actually find a matching like base, let's say, or matching event almost if the scores or if the normalized values are relatively close to each other. Uh, but here it's more about, I guess, how many signals that you have.
Yeah. So I think the event segmentation is probably a better alternative to match bonus.
Mhm.
So if but I think at that point when we tried event segmentation, it was not really good for slightly long
Mhm.
references. So we were like, and it wasn't working for our use case. Maybe the event segmentation has improved at this point. I think I was trying out the uncalled implementation of event segmentation. At that point, it wasn't really working for our purposes, and we figured we need a different strategy, a slightly smarter one, because we don't want to build a complex hardware just for
Mhm.
event segmentation. So I
Yeah.
think this worked to some extent, and especially given our expectation was that we don't need to make this work on a human DNA, right? We are searching for
Mhm.
pathogens, and we are limited to
Exactly.
the largest infectious virus. At that point, everybody was like, we need to look for viruses. So 100,000 bases is the length of the largest infectious virus, and therefore all our experiments are mostly limited to this length at this point. So I'm going to talk about how we moved away
Mhm.
from here, but at this point, I think the match bonus really worked well. It needn't necessarily work for very large genomes. That's something that we haven't explored.
Sounds good. Thanks, Harry.
Thanks, Sam. Moving on, so our implementation in hardware was a systolic array. So it's an array of processing elements. So you can see from 0, 1, etc. up to 2000, up to 1999. So all of those systolic array elements are connected through wires, and they are essentially simple processing elements. And on the chip, we have buffers for the query and the reference. For the query, we have a ping pong buffer. It's more like you write into one while you read from the other. And the reference buffer is sufficiently sized so that you can program in the largest non-infectious virus. And so you can see here that the squiggles are coming out of the DRAM, and these squiggles are written into the query buffer. And while the normalizer reads in another squiggle from a different query buffer, and this write and read can happen in parallel. And the normalizer does the normalization that I just talked about on the squiggle, and it passes this normalized signal to the systolic array, and the systolic array does the matrix computation. So this is our the green boxes, or the processing elements, are, think of them as along the dimension of the reference. So each of them stores a reference value internally. This is a one-time configuration. And then it computes the row dimension of the query dimension of the matrix in every iteration. So in every iteration, let's say in the first iteration, P0 does its job. The second iteration, P1 and P0 that it does its job. So that's how this computes the 2D matrix in each iteration. And finally, so if you look at some of the high-level features of this hardware, you can see that the reference buffer is on chip, it's programmable. And we also have mechanisms to write back from the last PE to DRAM. So this means that we can support variable query length, and therefore this enables more effective read-until optimizations. What this means is that sometimes a small prefix length is not enough for us to confidently answer the problem of selective sequencing. So we might need to look a bit more. So we might need to look slightly more to get a confident answer. So therefore, it's better to have this kind of feedback mechanism so that you can iteratively keep looking until you're confident enough to say that this is a target or not. That's why we have this mechanism in place. And if you look at the SOC for targeted pathogen detection, you will see that I think John just spoke about the software MinION that interacts with the MinION that comes with the ONT. The MinION dumps this information. I mean, if you connect MinION to Jetson Nano SOC, it is a SOC that comes with the GPU and a few different ARM cores. So that is all of this that we show here. And instead of the then we envision that on the SOC we do have SquiggleFilter ASIC. So ideally, if you have a portable SOC with an ASIC, and the idea is you can directly look at the squiggles from the DRAM and then provision a feedback mechanism to the MinION to understand and eject if the DNA is a non-target. And all the filtered reads that essentially from the output of the SquiggleFilter, all the filtered reads, they are alone passed to the rest of the software pipeline. So now we got rid of 99% or more of the input. We only need to look at 1% of the input, and this doesn't take more than a minute or so. So this entire pipeline runs in 1 minute, approximately 1 minute. And we run the base calling and variant calling and everything on the on the GPU and the ARM core, and it's a very, very small workload because we have this filtering mechanism in place. The small workload, and then we get the final results as to what variants exist in the SARS-CoV-2. So we'll be able to say if this is a different strain of SARS-CoV-2 or something like that, or so that's the whole pipeline. But then SquiggleFilter does this job of targeted virus or targeted pathogen detection. So if you look at how SquiggleFilter performs, if you look at the latency and compare that to GPU at Jetson Xavier AGX is the baseline, you will see that SquiggleFilter ASIC has a very low latency implementation as expected, right? We have a custom piece of hardware that is doing its job. And then if you compare the throughput, you will see that we do have a MinION output, the sequencer's output, and a projected output of the MinION, which is 100 times. This is the projected output in the future. We see that SquiggleFilter is capable of meeting the future output. So it's built to meet future demand. And it's outperforming the base caller running on different configurations on different hardware. I'm sorry, the same configuration. So it's Capri Lite, it's the same configuration running on two different types of GPUs. So high-level summary of SquiggleFilter. It is a programmable ASIC for new viral genomes. So, and it can detect viruses, can detect known and novel viral strains, which is an attractive feature in my opinion. And it has a very high throughput compared to an edge GPU. And it enables read-until optimization. And we measured by how much we gain out of using Squiggle-based selective sequencing pipeline running on SquiggleFilter and Jetson SOC. We saw 1.6 times lowering in terms of sequencing time and cost. This I feel is quite significant. Imagine lowering the cost of SARS-CoV-2 detection on the edge for all the patients in the world who might be suspecting if they have an infection. This I feel is a big win. However, at this point, we started taking a look at SquiggleFilter, and we realized that SquiggleFilter is built for the current problem, right? It was viruses at the time. And it can only detect viruses because of the limited on-chip buffer. And most bacteria have longer genomes, ranging in the order of millions. And therefore, SquiggleFilter wasn't easily scalable. Another problem that I'd like to highlight is ASICs are not typically democratized. It's not available through all over the world. And therefore, at this point, I talked to the NVIDIA CEO, Jensen Huang, at this point, and we initiated this whole collaboration where we implemented SquiggleFilter's algorithm on GPUs, and this is what we call Squiggle DTWax. So DTWax stands for Dynamic Time Warping Accelerator, and this is SquiggleFilter algorithm built on GPUs with optimizations in place for GPUs instead of ASICs.
Maybe before we move on to DTWax, there are some questions on YouTube. Maybe I can also forward them to you.
Oh, yes, yes. Sure.
So maybe one question is really about the difference between the signals and bases in terms of the space that they consume in memory or in storage in general. So which one is taking more space, and why is that the case? I think I summarized the question nicely, hopefully.
Thanks, Jan. So typically the signal would take a lot more space. It would approximately, in the case of ONT, take 10 times more space than the bases. And the reason is it's really hard for us to control the pace at which the DNA moves through the pore. And at the same time, we do need some kind of consensus for us to observe a DNA as it moves through the pore. So we don't want it to move too fast or too slow. And we have certain mechanisms that that the company has come up with to ensure it moves at almost at a steady pace of approximately 400 bases per second. So now that's still a lot of data. But at the same time, you have this 10 times redundancy in terms of sampling each base. So that's a lot of redundancy. And it's only in some instances that you need to retain this data. Almost in almost all pipelines, you don't retain this signal data. You only retain the base call data. But there are some instances where you might need to retain this data. For example, methylation detection is one such application. So and some cases where typically, let's say, in point of care testing in a hospital, it's sometimes it's a requirement to archive some of this data for a few years. So in those few instances, it might be necessary to retain this information. And I think to put things into more context, sometimes I think as we are seeing more recently, there's also this emerging trend with the R10 pores where it is actually generating some kind of junk data in the sense that it's combining reads into one, and sometimes you still need to retain this data, right? When you have a new chemistry and something is not working as expected, sometimes you end up retaining just to explore what's going on. So in all these instances, there is this problem of one being more than the other, but almost always otherwise you don't really care because you just don't care about the squiggle data anymore.
Yeah, that makes sense. And while we're at this slide, someone is also wondering, so this whole process is happening in real time. I
No,
assume it can happen in real time.
it can happen. It can happen. But I think everything until SquiggleFilter is what we envisioned happening in real time. And then once you filter off all the known targets, what remains is just the 1% or more, or 1% or so of the viral reads, right? And it's not even a challenge, and it runs quite quickly on the variant calling pipeline. So we wait until we have a certain coverage. So like the 20 to 30x redundancy on the viral genome. So once we have accumulated a certain number of viral reads, that's when we trigger this pipeline. And there's very little benefit from trying to overlap the first half and the second half, because the second half doesn't take much time. And so that's something that we haven't explored, but it's not worth exploring in my opinion, especially if the reference genome is really small, like a virus.
That's true. So I also have one question. This is a bit high level. So you showed this motivational slide where we had COVID and then PCR primers, and then they were developed and being used late, right? Yes, so this one.
Uh-huh.
And I guess you really motivated really nicely that if you had maybe a computational platform that can replace the PCR, we would perhaps be testing the cases much earlier.
Mhm.
Uh, so SquiggleFilter was not perhaps out at the time, or maybe not used very commonly, but Readfish was out. and we were aware of read until and nanopore sequencing and so on. Yet it was still not used. So I assume there were some challenges or some caveats of using it and replacing instead of essentially using PCR, right? So what do you think the challenges were? And did we solve these challenges, or how can we solve these challenges going into the future for the next pandemic, which we don't hope that it will happen?
Great question again. I would imagine there are multiple factors there. I can think of a few. So you're right, Ratefish was there. It just, I think, probably, I think it came out around the 2019-2020 period, right? The publication came out around that
Yeah,
time.
around that, yeah.
Yeah.
Read until
So
until 2016.
yeah. But I think it wasn't really integrated in the ecosystem. That's probably one of the main reasons I would think. Although maybe some of the research community knew that this existed, it wouldn't be something that they necessarily tried in their own lab. Secondly, I don't think it was recommended in any of the workflows that Nanopore said should be used for SARS-CoV-2. That's probably one of the second reasons. But from among the users that really used it, I know that there was this one particular challenge with respect to looking at the instances where somebody is really infectious versus somebody is very mildly infectious. And if you don't stick to the right, I mean, if you don't refine your workflow to an extent where you carefully extract the DNA to make sure even the slightest traces of SARS-CoV-2 exist in there, because you're not doing this wet lab enrichment, it's possible that you may not detect it in the pipeline itself, because it doesn't get through into the MinION. So that's certainly a specific amount of expertise is required in the extraction process and the sample acquisition process, and how you conduct the experiment to make sure some traces of SARS-CoV-2 gets into the pipeline, right? So imagine a person is really infectious. It's not just the virus that's going to be there. It's going to be a lot of human DNA in there, because a lot of human entities are trying to attack the virus. A lot of human DNA is really packed into that little space. So I have heard some challenges on confidently getting a 20 to 30x coverage on a SARS-CoV-2 virus, and they would be working on refining these workflows at that point to make sure they get some virus coverage. So I think these are potentially some of the challenges that I'm aware of. Maybe there are more, I'm not sure, but I know at this point there are a lot of publications that have established that this is possible, and they compare it to RT-PCR based studies. So at this point, we have working workflows. It's only a matter of adoption.
Yes, I completely agree. It's at least good to see that the field is improving a lot on this direction, and hopefully, library preparation will also be very easy
Yes.
without requiring perhaps expert effort.
Yes.
Okay, good. Thanks for the answer, Ari.
Thanks,
I
Sam.
don't have. Let me see YouTube again if there. Okay, I guess we don't have more questions on YouTube.
Okay, thanks for monitoring it for me. So I'll keep going. So after SquiggleFilter, which was a software hardware solution for pathogen detection, as I just talked about, so we moved to DTWax. So DTWax is dynamic time warping accelerated. So the difference is the SquiggleFilter algorithm is now optimized and running on GPUs. So you have Squiggle input coming in, DTWax running on a GPU would compare it to a target pathogen reference, and then give us an answer as to whether this Squiggle input is mapping to a target pathogen or not. So in case it is a target pathogen, as we know, we just sequence it. We don't need to take any action. But if it is a non-target, we need to eject the DNA out of the sequencer. So we need to inform the sequencer that this is a non-target and we need to eject it. And when we started out with DTWax at NVIDIA, while I was at NVIDIA, we realized that there was this gap with respect to the best possible implementation of a DTW on GPU versus the throughput of the MinION. So Cody DTW is work from one of our current collaborators, Bertil Smith. And Cody DTW does dynamic time warping, but for a different application. But we tried and repurposed it for selective sequencing, and we figured there's a gap. And that's when we optimized the algorithm to meet that cap. I'm talking slightly a little bit about the algorithm, and then we'll talk about the optimizations that went before we bridge that gap. So to start with, we understand how the SquiggleFilter algorithm was mapped as historically, right? On a GPU, how would you map this algorithm? That's a question that I learned during this process. So we can break the references into tiny segments, and then map one segment to a thread on a GPU. So T0 to T32 or T64, whatever is the number of threads in your thread block on a GPU. And to each of these threads, you map a segment of the reference. And then you stream in the queries as they come in from the lower thread to the higher thread. So let's see this in action. So the references are stored on the thread as segments. So imagine these references stored in shared memory, right? So it's each thread has access to the shared memory on a block, and that's when we store the references. And the query comes in from the global memory or the DRAM, and it gets pulled into the registers. And let's see how it works. In the first iteration here, the thread 0 computes all the 3 cells that it is capable of computing based on these references and this particular query. And then the second iteration, this query is passed to thread 1. And we make this work. We'll talk about the actual stuff that makes this work. This is called register shuffles if somebody is familiar with GPU programming. So you can actually stream this query from lower thread to the higher thread. So basically register to register communication. And then now you see along the diagonal, you have 2 threads doing the work. Next iteration, you have 3 threads doing the work. And at this point, you also need to stream in the diagonal cells value. So that's another register shuffle that we're doing. And we're skipping the left to right communication because we're assuming there's no deletion in the reference. That's again the same assumption from SquiggleFilter that we are making. So it's only the diagonal and the query that's being streamed. And the next iteration, you have all the 3 segments computing. Next, we have two of them computing, and then we are at the finish. So we computed the last row, and then finally we need to take a minimum and then compare it to a threshold to see if the error is within this threshold. If the error is within this threshold, we'll call it a map. And so a few questions to intellectually understand this at a high level. If we have query and references of lengths Q and R, how many cycles does it take in case T is the known number of threads? So the way to answer this is this. I hope just review it in your mind. Usually ask students in a lecture. So if we have R as a reference length, then we have T as the number of threads. You can imagine R by T is the number of segments, right? And then Q is your y-axis. So R by T, or rather the number of segments plus Q minus 1 is approximately the time that it takes to compute this matrix. So it's approximately the number of yellow segments plus the size of Q. And then we also need to compute the minimum of the last row. To do that, if you imagine a reduction operation on the GPU, it will take R by T, or the log of the number of segments amount of time. And is the next question is, can we get rid of this log R by T? So there is an alternative way to compute the minimum of the last row, and the way to do that is compute the minimum of each segment and pass it on to the next segment as the matrix computation proceeds. And how do you pass this on? You can use warp shuffles or register shuffles, which is register to register communication on a GPU. And this can potentially be overlapped with the matrix computation. So this log factor gets overlapped with this matrix computation factor, and that's how we save time. And another optimization that goes in here is, do we really store array elements like this with respect to the reference? So if you are more of a GPU programmer, you would be like, if every thread accesses R0, R3, and R6, they are like spread out in memory, right? Because if you look at this as a linear array, R0, R3, and R6 are not close together. They're not contiguously packed. So that leads to a lot of bandwidth underutilization due to several reasons. But is there a way that you can minimize this bandwidth and underutilization? I think that I'm trying to abstract this at this point. Yes. So if you pack R0, R3, R6 together, and then the threads are reading R0, R3, R6 together because they are closely packed in memory, not packed with a lot of space in them. So you can read these in a single read from the global memory instead of having to issue multiple reads over the DRAM. You can read this in a single read, and that is what we call coalesced memory loads. And this gives us a lot of benefit in terms of bandwidth utilization. And if you look at the high level optimizations in DTWax, DTWax is very optimized for data locality. So when I say so if let's look at the three different areas where we try to optimize for communication and data locality. So inter-thread communication is one place. So think of threads. Threads communicate in the register space on GPUs. And if you look at the prior work, only the query was kept in registers, but now we are keeping the query and also the minimum scores communication through registers. And inter-warp communication. So now you're talking at groups of threads on a GPU. Warp is a group of thread. And prior work communicated this output of the last column or maybe the minimum value. All of this communication was happening through global memory, which is a high latency communication, right? And we replaced this with shared memory. So we have a shared memory on the NVIDIA GPU, which took in the last column in case the reference is really large. And then we need to break this computation into submatrices. You could take the last column into the shared memory and then do the next iteration of the next submatrix. So we replace global memory with shared memory for some of the inter-warp communication. And finally, for the global memory loads, as I discussed while fetching this reference into shared memory, we ensure the global memory loads are coalesced. This was not ensured in the baseline work. So this is a pre-processing technique that we did just because we knew how the virus looks like. So since we knew the reference genome ahead, we went ahead and used this piece of information to reorganize the Squiggle reference. So we implemented an optimization to improve the GPU occupancy, and we minimized the branch divergence using several CUDA programming tricks. And this helped us easily cross the throughput of the future MinION. And we also, I think these are some of the strategies that went in. So kernel tuning and asynchronous data transfers between host and device. So we try to maximize the GPU occupancy by tuning the kernel, and then also helped improve the GPU utilization with asynchronous data transfers between the host and device. So overlapping compute with data movement is something that we tried. And then for minimizing the divergence, we did loop unrolling in especially with respect to computing the first submatrix and the last submatrix in a really long reference. The first and the last submatrix computation and score updation happens differently. So unrolling the for loop gave us some benefit. And finally, we realized it's important we keep pushing higher and higher because if you're using a GPU, might as well make it as performant as possible so that you can pack more work on it. So what we did is we went ahead and improved the throughput and latency with FP16 vectorization. So at this point, we were using FP32 and then we moved to FP16. So that's approximately 2x the throughput. We observed 1.7x throughput gain here. And yes, I think it's at this point that we went ahead and did the pre-processing of the reference to enable more coalesced memory accessing. So remember I told you about reorganizing the reference squiggles so that memory loads are coalesced. So we went and plugged that in. And then finally, on A100 GPUs, we realized there's a way to use this unused piece of hardware. So remember, I think when you are given access to a large piece of hardware, you should definitely profile and see what's going on. So at this point, we were really going towards the compute bound limit on the hardware, but we weren't really there at that exact point. So we realized there was this piece of hardware that was sitting there being unused. That was a tensor core because you're not doing any matrix multiplication. But on A100s, there was a way to fool the compiler by formulating A times B as A times B plus C. So it's like faking a fused multiplier out of a multiplication. If you can fake that, and either there was a way to use the compiler to translate the programming to an instruction that led us to utilize some part of the tensor core, but for non-tensor operations. And we also used highly optimized backend libraries like ODNN for normalization. That was another integration that led to a small performance win. And we still relied on SquiggleFilter's underlying assumption with respect to the cost function. So that's at this point that we chose to skip the reference deletion under the assumption that reference is a golden truth. We don't expect to see any errors in them. And SquiggleFilter had already shown a robustness study with respect to how this error could affect our final answer, and the effect was very, very negligible. And finally, we did compare to some of the state-of-the-art works at that point. So DTWax, again, the evaluation was performed for speed up and speed up to cost up, because we were doing this on a cloud on an A100 GPU, which was very hard to access at that point in time. And DTWax performed very good in terms of speed up, approximately 2x speed up and approximately 4x speed up to cost up. And DTWax could meet future sequencing throughput as well. So if you see the green line, that's a future MinION. DTWax had a really good throughput that it could meet more than what the future MinION could hit, but at the same time, also give you a very small classification latency at the granularity that we were processing. And DTWax at that point was the most accurate solution for viral genomes. So we got up to 90 plus, I think close to 95-ish F1 score, and this is our confusion matrix for DTWax versus a few of the other tools that existed at that point in time. Right now, I think the space is more evolved. I think Jan and Onur's lab has more tools. And then there are also a few tools out in the community that were inspired by DTWax and SquiggleFilter. So I think this set of baselines would, if one were to do the experiment today, it would be a lot larger than this. And finally, if you were comparing DTWax versus SquiggleFilter, it is obvious that SquiggleFilter is an ASIC solution, so it's supposed to be more area and power efficient. And but DTWax is built for throughput. It's running on a GPU, so it's no surprise that the throughput is approximately 2x higher than SquiggleFilter. And we didn't stop there. Right now, we are repurposing DTWax for a very innovative application. Professor Jeff Nivala at the University of Washington, Seattle is trying to identify protein sequences and sequencing them for the first time on a MinION. And DTWax is being repurposed by their lab to identify protein sequences for the first time in the world while they're being sequenced. So I feel it's important that as budding engineers / researchers, we all try to keep our tool active, because sometimes I see a tool being developed and it being left there, not being used anymore. But if you can make it more reachable and make it usable for different applications, I feel that's a big contribution. This is something that I actively try to do. I see a hand. Please go ahead and ask your question.
So hello, thanks for the talk. I actually have a question about the previous slide. Maybe, okay, just one more slide back, if you could. Thank you. So I think you mentioned that DTWax is the most accurate solution for viral genomes, right? Up until now. So how about bacterial genomes? Because like I think for the pathogen detection, you already mentioned the bacterial tests as well. Could you also like try the bacterial genomes?
So that's a great question. I think the genome that we tried here is a lambda. It's actually a bacteria. It's one of the bacteria. But that's a great question. I don't think we explored that yet. But that's a great question. And there's nothing stopping you from exploring that. But I think at that point, what we were doing was a one-to-one comparison with a SquiggleFilter. And I think that was mostly the direction that we wanted to go in because of SARS-CoV-2 and all the highlight on that. But lambda is in turn a bacteria. It's actually a proxy to the longest virus. So it's really hard to get a sample of the largest infectious virus. And it's not commonly observed. So lambda is a proxy. It's actually a bacteria. It approximately resembles the size of the largest non-infectious virus. But it's a great question. I feel that is something that is worthy of exploration. But at that point in time, the DTWax paper itself was written for pathogen virus detection. So that was a whole motivation, and hence the study was limited to that scope. But I think after doing all this, I look back and I'm like, okay, it's worth exploring DTWax for bacteria as well.
So related to that, maybe like I'll ask what do you think like the design would be, or like what do we need to change the design into? So for example, for the bacterial genomes, in the previous slides, you said that for the memory allocation in the GPU, like shared memory is heavily used, right? And if the reference it cannot fit into shared memory, you mentioned that the reference is, or like the matrix is actually like divided into subparts, as far as I could get from the talk. So like if you think about the bacterial genomes, again, related to the future work of the DTWax, I'm sorry, then the shared memory is, again, the size of the shared memory is going to be a big bottleneck for that. But like, do you have any, let's say, design decisions in mind, or like something that you can maybe find a solution for that problem? Because usually, like this size of the shared memory in GPU, let's say, computations is a big problem in that sense.
So that's a great point again. So I would imagine, although DTWax is programmable to larger reference, so at this point there's nothing stopping you from using larger reference. It would automatically scale to larger reference. But the idea is it is indeed a costly piece of accurate testing with respect to a human genome. And I think Jan has beautifully explored with DTW for his, I think, row align backend, if I'm not wrong. And that is, we are potentially exploring that collaboration. So not using DTWax, which is kind of like a high accuracy testing, but expensive on all the scenarios, but limiting it to those scenarios where we suspect a match. So think of more like a Minimap2 backend, where you don't do Smith-Waterman. If you're familiar with Minimap2, you don't do the costly piece of operation every time. You only do it when necessarily to test all the suspects that you think would potentially be a match. So I think at this point, although we could make DTWax work easily by using a DGX cluster or an AMD box of GPUs, like you could make it work on a bacterial genome. But the more efficient solution here would easily be to have a front end, more like a seeding and a chaining strategy, and then potentially use a GPU accelerated DTWax to map for bacteria. That is an active project that I'm exploring with the Jan for a different purpose, but that's underway.
Okay. Thank you. Thanks a lot for the answers.
No worries. So moving on. Okay, so we discussed DTWax as being used for this application right now, and I also brought up that we are looking into how we could add a front end to the GPU accelerated DTWax, or better do accelerated alignment on long references for certain niche applications, which we're still working on. So I don't have slides on that. Finally, I'd like to highlight some of the work that we did in the base call space. So all of this time we were talking about alignment in the squiggle space. Now we are pivoting to the base call space. And why is this important? Because we have Minimap2, which is an approximate string matcher in the base call space that is used in many applications, especially in in all of the pipelines that use that are used with MinION. Minimap2 comes up. So there are two important contributions that I'd like to highlight here. One is MM2AX and the other is MM2GB. MM2AX is the first solution for accelerated Minimap2 that was able to do 100% accuracy match on a GPU. So MM2AX was unfortunately closed source from NVIDIA. And later we at AMD came up with MM2GB, which is an open sourced accelerated Minimap2 that runs on both NVIDIA and AMD and has added optimizations on top on it. I'll briefly discuss how we use this in cancer detection in a pipeline at the University Hospital in Basel. So hold on. Minimap2 is the step after the base caller. It looks at the reads and then maps it to a target virus genome and then tells you where these reads map to in the virus genome. So it is MinION 2 again is a high accuracy long read aligner. Let us take a look at the MinION 2 aligner and see how it performs on a CPU. So if we were to profile a MinION 2 on a CPU, and with respect to the reads, so let's say the output of the ONT MinION is very variable. That's something that probably I should have highlighted before. The length of this reads varies a lot from experiment to experiment. There's a lot of uncertainty with respect to exactly what the average length observed would be, but there are certain wet lab measures to make sure you get small reads versus very long reads on an average. But then let's profile MinION 2 in different situations when it's small, somewhat larger, and very large. And you observe that the fraction of time spent in a particular step called chaining in MinION 2 is really high. And we identified this as the bottleneck in MinION 2, especially for ONT reads. And chaining, primarily we detected that it exposes less parallelism. So it was a really hard job if we were to parallelize MinION 2 on a GPU. And the other factor is irregular workload and control flow. So as you know, the output from the sequencer is very irregular in terms of size. And again, this in turn leads to the fact that the input to the chaining step is also very irregular in size. And the control flow in the chaining step in MinION 2 is very irregular. It has a lot of if statements if you look at in the code. So it's not the same amount of instruction that gets executed for every piece of chaining that we need to do. And this is the high level result before I jump into the optimizations that we did. At NVIDIA, we developed MM2AX, which is now sold, and a package is sold as part of their MinION 2 beta offering. I think it's no longer a beta, it's more like a stabilized product at this stage. So we were able to not only extract more parallelism out of the chaining step, but also accelerate it on the GPU. And across the read lengths, we observed different speed ups and speed up to cost up, partly due to the fact that the workload is very unbalanced. So it was difficult to get a steady performance gain, but we did measure a significant speed up on GPUs. And this is a summary slide again on all the optimizations that goes into MM2AX and MM2GB at a high level. This is more of like a introspection, right? So you do MM2AX and MM2GB, and then you look back and analyze what all went into the effort. I realized there are probably 4 different buckets. One is algorithmic transformation. So when you are using a software in a clinic versus using it for research, it's a very different scenario. You just cannot compromise on accuracy. It's a period there. So you can talk about all the optimizations that you do in hardware, but a clinician would just not be okay with using it if it is not able to replicate the results that he's used to seeing on the screen, or it should be better. I mean, if it is worse than that in terms of accuracy, the clinician is not going to accept that. So MinION 2, when we accelerated that as MM2AX, that's the number one thing that we considered. We have an exact 100% match with respect to accuracy. And at the same time, we did some algorithmic transformation to ensure we extract more parallelism onto the GPU. And secondly, we exploited the sparsity and leveraged locality. So the idea is MinION 2, although it is very irregular, chaining is not performed on all the seeds or all the input elements that are passed to the chaining step. So this is something that we could condense. So we could remove the elements in the array that where we didn't really have to do any compute on, and just don't transfer them to the GPU. So this saves a lot of communication burden. And then leveraging locality. So the idea is we realized there were instances when a group of neighboring elements would be operated on together. And I'll have a slide on this, but then we prefetched this group of neighboring elements onto the GPU ahead of time because NVIDIA has a hardware prefetcher for L2 prefetching. And branch divergence. So there are a lot of CUDA tricks that helps you with minimizing branch divergence whenever possible. So the idea is you can compensate for branch divergence by adding in a lot more compute. So put in a lot more compute there and get our branch divergence as much as possible. And workload balancing. So this is a bunch of our optimizations that went into MM2GB mostly. So we did binning. We had some binning in MM2X, but mostly MM2GB too. Idea is group the ONT output reads that are similar in lengths together and process them together so that the GPU has a somewhat balanced workload to operate on. And then next is sorted inputs and prioritized scheduling. So with MM2GB, we actually not only binned the reads, we also sorted the reads. And we prioritized the scheduling for that. So the idea is we should start working on the longest read as early as possible so that it finishes by the time that you process all the other reads. Because that is your tail latency, right? So your longest read is essentially a critical path. So unless you finish that, you are not ending the processing. So you need to start on your longest read as early as possible. And the sequence cutting, the idea is whenever you have a long piece of work to work with, you try and break it into smaller chunks if possible. So this helps in better workload management. Custom GPU kernels, right? Essentially writing custom GPU kernels for different sizes of the workload. So for a short workload, write a different kernel. For a medium-sized workload, write a different kernel. For a very large workload, write a different kernel. That's the idea. Let's jump into the details now, but I'm just wanted to put the high-level summary there. So first, how do we do this algorithm transform? So remember, I mentioned chaining is a sequential step. So at a high level, you can see that this array element needs to be compared to, this array element and this array element needs to be compared to using a score generator and then the previous value at this array element. So the comparison happens between these two and the updation happens in the direction of the arrow. So if you see the direction of the arrow, that's how the final updation happens. And then the next step, you need to do this comparison, A1 and A3 score generation and comparison and updation. So again, see the sequentiality here. It is only after A3 is updated in the first iteration that you can able to compare with A1 and then update A3. So this sequentiality is inherent in Minimap 2. And we call this algorithm the backward algorithm in Minimap 2 because this comparison and updation happens in the backward direction. But we did this transformation to a chaining algorithm. What we did is we transformed this backward step into a forward step. But we made sure that the output is exactly matching. We ensured a few changes to the algorithm. Some of these changes are just in the way the values are compared. But essentially, we made sure the final output is unaffected. And this was critical in having MM2AX or MM2GB adopted in a clinic. And this is what we call the forward transform because now the comparisons are happening in the forward direction. And as you can see, there's no sequentiality here. That's a good news. So all these comparisons are independent of each other and they can happen in parallel because updations are also independent of each other. When you're updating A4, you can update A5 and A6 in the direction of the arrow. So they're all independent. And one other optimization that went into MM2X is identifying the sparsity and condensing this sparse workload vector. So we realized that 67% of the times, the elements in the array were not really operated on for chaining. So we just stopped transferring them to the GPU. It's as simple as that. Don't transfer data that you don't want the GPU to be working on because you will be wasting a lot of bandwidth, the PCI bandwidth in doing that, and also wasting a lot of time. Just don't do that. And then finally, we observe that when the chaining is being performed, I know I haven't gotten into the details of the algorithm. I don't think it's relevant, but we identified that a certain number of elements could be prefetched together onto the GPU for a warp to operate on. And you can benefit from cache locality in this case because the data is already in your cache. And we were not in a position to use shared memory at all because the algorithm itself is very irregular. And programmatically prefetching fixed size data into shared memory wasn't giving any benefits because the reuse amount was very small. All we could do is like plan for fetching a fixed size of data so that it at least gets to be in the cache. And we observed that yeah, I think we observed that at 32, we were close to like getting a good enough, like a 95%. I think 95% is the number of scenarios where prefetching a 32 number of elements gave us benefits. And the other optimization was we realized the input workload is very irregular, very imbalanced. So we went ahead and binned this workload right at the base caller itself. So base caller, remember, knows the length of a read as it base calls. And if we could use this information to just bin them, we're not really sorting them. So it's an order of one operation. So you're just binning them, sending the read to the right bin based on the length of the read. And then we combine very few long reads with multiple small reads. So if you look at the distribution of OINT reads, I realized maybe I should have kept a picture. There's this long tail for OINT reads where you have very few reads with very high lengths. So and this is what comes to the critical path of read alignment on the GPU. And we combine these very few reads with multiple small reads so that the GPU is occupied. So we don't want just one CU or SM on a GPU occupied. We want all the CUs and SMs to be uniformly occupied. And this combining strategy was very critical in MM2AX, especially for very long read lengths to get high performance. And so of course, we tried minimizing branch divergence. I think partly the forward transformation helped with some branch divergence, and then we traded off compute for divergence. So I think I got this other way around. So unified early termination check, unified false ladder to a single line. So these are all tricks in CUDA where you can add more compute to get rid of divergence. And using warp synchronized intrinsic. So wherever you can use a warp synchronized intrinsic and avoid async threads inherently, you can use those again a CUDA trick. And now I'm pivoting to MM2GB, which is our open source implementation of accelerated minimap 2 from AMD. And this is what is being used in the clinic. So here we have 3 more optimizations that I would like to talk about. So we realized that with MM2AX, the workload that chaining was processing was still irregular. I mean, even though we tried a few optimizations in terms of condensing the workload and then prefetching some of the elements that we think were processed together and did that algorithmic transformation, the workload itself was still irregular at a fine-grained level. At a coarse-grained level, we kind of binned and took care of the irregularity, but at a fine-grained level, the workload was still irregular. So what we did here is we realized that we have the sequential processing of a read, and then there may be instances where when you're chaining and there's no dependency between 2 small segments on a read. And so when we identify instances where there's no dependency between the 2 small segments, just cut them into small chunks. So we do a cut here, and we do it inside. There were many instances where we could actually do this cut. So this kind of ensured that we get much smaller segments than one big segment, one big read to work with. And this cutting actually helped us better parallelize. So instead of having a few segments for the GPU with hundreds of CUs or SMs, we can have multiple smaller segments. It's better workload management essentially. And the other idea was, although we have some kind of fine-grained workload balancing at this point, we needed specialized kernels for each scenarios when you have a very long workload, small workload, and a medium workload. The idea is when you have a very long workload, we realized there's more compute being performed, so we needed more threads there. But when you have a small workload, there's less compute being performed, so we needed less number of threads, and that was being more performant. And the other idea is making sure your critical path is no longer a critical path. I mean, so the way to ensure that is previously the longest segment would be your critical path, but if we can start processing it early on, we can overlap that with processing multiple short segments. That's the idea. So prioritize and balance workload automatically. So we would schedule the longest piece of segment first and then go to the shorter one. So it's in the descending order that we would schedule. And then yeah, I think this kind of shows us that with this prioritized scheduling, we actually have almost all the blocks doing some kind of processing together versus like some of the blocks processing and some of them not processing in the default case when you don't have any prioritized scheduling. Finally, MM2GB, it's a win for MM2GB given the fact that it is used to make in-operation room decisions in a hospital at the University Hospital in Basel. And the idea is a neuropathologist there is using MM2GB running on a piece of hardware on the edge, connecting it to a MinION, And then he's running in a very low power, very privacy important and privacy critical environment where you have to do this processing on edge, on the edge with no feedback to the cloud or whatsoever, and in a very low power setting. And MM2GB worked right out of the box for it. So MM2GB works on NVIDIA, AMD, and it just worked on the edge for him right out of the box. And he's using it for lymphoma detection. So essentially, a surgeon in the other room would send him a brain tumor biopsy, and then he would be able to sequence this biopsy in 20 minutes versus a month of sequencing. Typically, a biopsy would be shipped to a lab to get sequenced. So it's a great improvement for him. And M2GB accelerates some of the compute or for him and spares the CPUs for him to run some other job. So he gets the answer in I think including the wet lab 20 to 30 minutes. And although the compute only takes sequencing only takes 1 to 2 minutes, so the wet lab takes most of it. And then he delivers his answer to the neuropathologist, who would then decide to whether he should operate the tumor, I mean, remove the tumor out, or it's okay for the tumor to stay in because it's not cancerous. So MM2GB is making an impact in a hospital real time as we speak. Conclusion. MinION, although it's a fancy revolutionary sequencer, which is easy for adoption, it lacks the compute power to work with. We identified a scenario for pathogen detection. We talked about SquiggleFilter. It was a custom software design, working on a custom piece of hardware. And we observed a very high throughput. And then we talked about how we could democratize the DTW algorithm by building a DTW solution accelerator on GPUs. And DTWax is open source through NVIDIA. And we got some nice speedups. I talked about how DTWax is used for protein sequencing at this point, and also how we plan to work with Jan to help deliver DTWax to more scenarios, maybe for larger references, and still have an accelerated performance. And finally, we talked about Minimap 2, how important Minimap 2 is in all the ONT pipelines, in fact, and how MM2AX was instrumental in delivering 100% accuracy for accelerated Minimap 2 on the GPU, and how at AMD we added on to MM2AX and open sourced it, and getting a win in terms of having it run on both NVIDIA and AMD, and additionally it being running in a clinical setting in a hospital, detecting cancers, saving human lives. And I think these are some of the DNA alignment works that I talked about today. So we started off with skew space alignment, and then we moved eventually to base call space alignment. And that's all for today. I'd like to highlight some of the publications with respect to these topics that we discussed. So we have SquiggleFilter, DTWax, Minimap 2 publications, and then finally we have the importance of genomics, which I worked with on. So yes, I think this is all for today. Thanks everyone for listening in. Please feel free to ask me questions.
Great. Thanks a lot, Hari. So maybe while people are thinking about their questions, if they have any, I'll ask a few.
Okay.
So first one is about MM2GB. Actually, in general, Minimap 2 acceleration. So yeah, actually, this slide is great. So here you have this max distance X, but in order for you to recognize this, you need to sort the anchors, right? So I'm curious how much time the sorting is taking overall in chaining, or maybe overall in the mapping part, because we saw chaining as one huge chunk. But I'm curious how much of it is taken by sorting, and are you thinking of like alternatives to sorting if it is a computationally expensive step?
So I don't think it's taking a lot of time on the GPU per se, because we actually split it into two. One is score generation, and then we do the chaining. So I think from the score generation, it is kind of after that step, it's kind of clear that what do not pass the score. So we really don't do any sorting is in this step. So this step is kind of clear, but it is actually in this step the prioritized auto workload balancing. It's here that we actually have to sort. And this is a very, very popular or very well explored kind of sorting mechanism on the GPU. So it's primarily looking at the length of the workload, and which is already known. When we are chaining, we know what the length of the workload is, and then we need to sort. And this is we do have well explored libraries both in CUDA and HIP for HPC applications. So this is a very well known strategy in HPC where you sort and then feed the work onto the cluster. So I would say it's not something that we are concerned with, because if you look at the critical path in the I mean, if you really look at the time, that's humongous compared to the time that we spend sorting. So this is a real deal breaker, I feel, especially compared to how MM2AX did this naive strategy. I feel this is a much better strategy, because you don't always have the base caller to bin the reads for you, right? Sometimes you're just reading base call reads from somebody else's folder, and then you are doing minimap2. So this is good to have, because you don't always have the base caller to bin the reads. So I think this is worth the investment, and irrespective of this, you do still get a win. And I don't think it is the big bottleneck at this point. The big bottleneck would be the rest of the pipeline. So that's something that we're working on. We do still care about the backtracking in chaining, the Smith-Waterman, and the seeding, which is memory board. So all of this needs to be moved on to the GPU, because the communication is the bottleneck at this point and not the sorting. You need to move data from the host to the GPU and get it back data from the GPU to the host. And we do have several initiatives here trying to put different pieces of this software. It's ultimately culminating in a GPU map to minimap2, all of it on the GPU. So that is the bottleneck. I feel all of it has to be on the GPU.
Yeah, that's interesting to hear, because probably I was biased that I asked the question about sorting, because this is what we saw for raw hash on the raw signal part. So when you do chaining over there, a larger portion of the time for chaining was actually spent for sorting, where like we were using radix sort similar to Minimap2. So I was perhaps assuming that maybe there's a similar trend over there. But yeah, thanks for sharing the insights. So the second thing about this, so earlier you mentioned that, which I agree, that you want to match the accuracy to Minimap 2, right? This is really important. Uh, but maybe another direction is, as you make things faster, maybe we can be even more accurate than Minimap 2, because Minimap 2 is also restricted by certain parameters, right? I don't know, maybe let's say, did you think about this direction? Like, are you even like shooting something higher than what Minimap 2 can do? And what your thoughts about these?
It's a good direction. I had this talk with the developer Professor Hengley, and I think our consensus was like unless there's a real need to do that, I don't think he was more inclined to do that. And I think my goal was simple, look at what the state of the art tool is, and see if there's a systems problem and try and improve it. But I really haven't focused much on aspects where there is a need for improving the accuracy. But I think at that point, it's a question about how better it works. I mean, the challenges would it be adopted as a state of the art, and is it worth profiling and optimizing it at that point? Because you really want to make sure something that you're optimizing for is a state of the art, right? So I think it's a good thing to have, but if I were to do it, I would always involve make sure that it's recognized by the developer of Minimap2, because it gets adopted, and then it makes sense for us to optimize it, because otherwise it could be a lost cause, I feel. So that's my thought. And definitely there are a few instances we do see Minimap2 getting improved every few quarters. So there are instances. So I think if one were to come up with such an instance, I would first make sure a PR is submitted to Minimap2, and Minimap2 has that integrated. So it becomes kind of like the state of the art, and then we could optimize it. That's my thought.
Are there questions from other people? Maybe I can check YouTube in the meantime. Okay, there are some, but I guess some clarifications about some details. Maybe we can take these offline. So I have one more question though. So this is again a little bit high level, which is mainly about the raw signal analysis versus the base code analysis. So what is your take about the future of raw signal analysis? Essentially, so what we see is maybe the base code analysis is in terms of accuracy is still providing perhaps a better accuracy for certain use cases. And the devices, the processors or the GPUs that we're using, they're also getting faster. So maybe they don't have much issue with the throughput of a data generation of a single nanopore, but maybe there are like different problems. So basically, I'm curious about what is your take about this? Where do you see the raw signal analysis and real-time analysis of raw signal without base calling will be in the future compared to the base code analysis?
Got it. Great question again. I feel maybe 20 years down the line, there wouldn't be any raw signal analysis. It would be, we would reach a point where the base caller is so smart that it would be able to detect all kinds of methylation and any kinds of epigenetic signals that the normal base calling traditionally wouldn't be able to. But I feel in the immediate term, if we were to understand some methylation patterns or any epigenetic signals that typically your base caller is not trained to because of the lack of data, I feel in the immediate term, raw signal analysis might actually get a short boost in terms of the necessity to go with it, just to understand more information from the signal. So I feel it's like a hybrid existence, I feel, in the immediate term. And I think it's important that we push for it because we better understand the signals to start with, to understand what the base caller is losing out on. And maybe potentially also, I mean, synergize the results from the base caller. I mean, there are different pipelines for methylation detection and getting to understand all epigenetic markers. So maybe in the immediate term we can have a win, but I think in the long term it might just disappear, and it's in our favor to disappear because retaining these signals is in fact a lot of it takes up a lot of space, right? It's not it we don't if the base caller is smart enough to avoid that, I think it should be doable. And I do get the feeling that that's potentially where Oxford Nanopore is headed as well. So I think in the long term, we might reach a stage where that's the situation. That's my feel.
Yeah, I think that makes sense. Sorry, maybe this will be a part of only a future engineering for future base callers by doing some analysis
Yeah.
between raw signals, and then you just provide as input
Yes.
to, I don't know, maybe it's some large neural models or whatever it's going to be using down in the road.
Yeah. I think we have a similar story in protein sequencing at the moment. We don't have data to train protein base callers for protein sequencing. So it's important we use techniques like DTW for understanding protein sequences. And once you have the data to understand those proteins, you could train a model and work with it. So I think there is always this use case for raw signal analysis at some point when something new comes up. So maybe in the future we are talking about metabolomics, right? Understanding about I mean something more than proteins. So maybe whenever you have a new type of data that you're dealing with and you don't know the underlying model or how to use AI on it, raw signal analysis will always pop up. So I think it's good to have a utility for raw signal analysis so that we can always use it when you have a new application. That's still going to persist. I think we are seeing that pattern coming on and off. So that's still going to exist.
Makes sense. Great. So I assume we don't have more questions. I guess then with that, thanks again, Hari. It was really great hearing really great works from you. So some definitely inspired also us. So I hope we'll keep collaborating also in the future. Thanks again for
Thanks for
giving a talk
having
today.
and organizing this very beautifully. Looking forward to our collaborations in future.
Same here. Then have a nice day for you and have a nice evening for the people in Europe.
Have a nice evening, everyone. Bye.